Hello, everybody, and welcome to Biochemistry 5631, Statistical and Computational Analysis of the Human Genome. This class, I'm going to give you a quick overview and introduction in this lecture of the second iteration after last year, which went really well. We've revamped it a little bit and just want to give you a quick overview of what the course is all about. And really, I like these little posters we made because I think the main goal of this class is for anybody with doesn't need any computational background to be able to go out and access data from the internet that's freely available while using best practices of Markdown and making sure about reproducibility at every step and documentation. But really learning the basics of data sciences and how to apply them to biology. And we like to do this in an inquiry-based way where we download a bunch of data from the Encode project about the human genome and analyze it. And I'll get into that as we go. And really, I want to first introduce that I'm co-teaching this with Michael Smulligan, who did an awesome job last year. He's a very, very talented computational biologist and data scientist and statistician, and we'll be exchanging videos. I'll be doing some lectures, he'll be doing others, as we both agreed that it'd be silly to have two people sitting here in a video while giving a lecture. So this is a meta plot of our last year's class and all the time we spend together pre-cooking the classes, which I'll get into as well. So I think we've got a better iteration this year and we're really excited. So let's find out what it's all about. Really, I think to start with, I'm just going to go on a high level here and make an analogy that if you remember woodshop class, I don't know if they still exist, but they did when I was in school. And you go into woodshop class and you learn a bunch of fundamentals, you learn how to use a bunch of different tools, and eventually you would make a gumball machine, usually, or something. And yet you couldn't build a gumball machine right away until you learned how to use each of the tools to build a single piece and then put the pieces together. And then there's usually some tricky parts where you have to get like the lever to get the candy out of the jar there. But in that same way, we're going to use Unix as really a fundamental platform, or Bash, I should say, for this class, the born again shell. We're going to use our Fiji servers, so everything will be done online and can be documented. I'll get into that as well. GitHub to record our data and collaborate there. And Nextflow is just amazing new revelation in data reproducibility and data sciences that the experts have made all these pipelines already. And so they've assembled them into a nice bundled system where you can now take internet directly from the data, push it through Nextflow or SnakeMate and these other things that organize pipelines, and you can use servers online to do all of this. So really, essentially, you can take free data, put it through an expert-defined pipeline, and get the data out that you need to then analyze. And so by the end of the class, we hope that everybody will have their own gumball machine. You'll have a presence on GitHub, on the Fiji servers here at CU Boulder, understanding Nextflow and all their tools and how to go get data. Most importantly is how we'll start the class. This is how do you get data and how do you know it's the right data? How do you know the file that you downloaded was the one that was uploaded? So that's kind of the analogy. And I hope everybody will get a gumball machine by the end of the class. So what this class will provide is we'll start with lessons on Bash or Unix, just the basics, because it's a really powerful way to move data around through different servers, actually. And we'll get into why that's important. In the next video, I'll show you the resources we have for how to learn Bash. How do you learn Bash? Do you read a book? What do you do? And this class is inquiry-based. And so we want you to learn Bash in ways that you will need to know them and actually practically know them to do data sciences and biology. And we'll get into that in the next lecture with the resources and supplements to this class. We don't want you to know the math and everything of all statistical principles, but we do want you to understand intuitively what statistics are important for what kind of data, what kind of null distributions do we have, how do we make null distributions, can we how do we think about the data statistically in an intuitive way? And then what are the tools and resources to code that? And we'll be doing that because R is based in statistics, essentially, or its fundamental thought process is kind of statistically based. And so we'll teach you basic and intermediate skills. Again, in the second lecture, I'll show you how we'll have lectures already pre-cooked and online resources that you will be able to just read the files with Markdown. And essentially anybody can take this class without being in the class. And then we're gonna apply all this information into an inquiry-based learning experience or application to analyze Encode big data. Right? Big data is kind of a misnomer, and this is not really big data because it fits in RAM, but it's big data as it's used typically. And then we'll spend a few weeks, as we did last year, what was absolutely an amazing experience of analyzing this data. We found something bizarre, and we as a class worked on it in an inquiry-based way to figure out what was going on with something. We even wrote a paper together and worked on data visualization, how to represent, how do you explain your results, your logic process, and then how do you present that visually in figures. In fact, we did submit a paper that got great reviews. I'll show that at the end. So it's really at least somewhat of a initial framework that works. When you learn a few of these things, you can learn more on your own, and it gives you kind of an intuitive way to go ahead and analyze the data of your choosing. But what we will not be able to provide, unfortunately, is Python skills. We know how popular this is, and Python's important. And we actually feel that by learning Bash and R principles, you'll be able to pick up Python and no problem after this class. Or maybe you already have it in your skill set, and this will be a nice supplement based in R. Again, just with the fundamental notion of how R works with the statistical intuition. And so we will not be doing anything in Python. I know it's very popular and it's a great skill set to have, but this class is more academic based in statistical principles of how we'd analyze the human genome, which R is almost ideal for, not to mention the stunning visualization you can do with R. We'll also teach you that. We will not be doing personalized analysis of your data, unfortunately. That's a nice way to do a class where students bring in their own data and we all work on it, but we like this flow of inquiry-based learning where everybody's working together on a new project. And the other thing is, this is not a help desk for outside class or help with your project. Nonetheless, we understand how important these things are, and we think after the second iteration, the third iteration, we could expand in these ways to actually include this inside and outside the class. So stay tuned for that. But in the meantime, this is what we won't be doing in class. So the class is kind of outlined into 6 modules. They're very rough and they're very integrated together. They go back and forth. But module 1 will really hit some Bash basics, concepts. So you'll feel really comfortable on a server of any type, Amazon, Google, really all will use a lot of these same things to get to them, to talk to them from your computer. And we'll use practical examples in how to do that. And then we're gonna get data. How do you go grab data? Where do you get it? How do you get it? And what happens if like half the file downloads? How do you know? And we're talking thousands of files we're gonna download. How do we know if we got the right data? Is that reproducible or not? Because what if we got 2/3 of the file or 99.9% of the file and we missed a little bit? We're going to get into how do you go grab data and make sure you have good annotation, use the best practices to make sure the data that was provided is the same as that you've received, and many other aspects on how to use APIs and other things to get lots of data at once is kind of the goal here. How to use basic Bash and go out and get all the data you want, but also checking to make sure it's good. And module 3, we're going to start talking about pipelines. ChIP-seq, RNA-seq, ATAC-seq. We'll focus in this class on ChIP-seq, RNA-seq, Pro-seq, chromatin, histone modifications, and other data analyses. Once we get one type of data down. We'll then expand to others later in the class. But we'll use these pipelines that are already built. And the cool thing about these pipelines is they're going to have stacked programs you've heard of, DESeq or MAX, peaks calling, or biological ways of understanding DNA binding events or RNA abundance in the cell. And we'll get into all of that into lectures downstream. And so the biology is not really important at this point. But the pipelines are all there for any type of seq or genomics you do. And we really like using Nextflow. They've got like really great pipelines. And the nice thing is also they're marked down at every stage of what failed, QC, and really nicely bundled and version tracked such that anybody can rerun your data or this class and get the exact same results and figures. Then we're going to get into some data analysis. What does ChIP-seq mean? What does RNA-seq mean? What can we learn about the human genome by looking at these different aspects? And we found something quite surprising last year and wrote a paper on it, which I'll get to at the end. But I think this is a really fun way to do an unbiased, large data set inquiry and see what we see and work on what we want to work on together as a class. And that turned out to be quite fun last year. So my model 5 is where we'll bring in a lot more statistics and we'll have pre-cooked exercises on all this. See the next video for the resources and how we're going to teach all of these classes in our Markdown with the goal of everybody being able to take this class and get the same results. So we're gonna look at the data and we're gonna ask questions about it, like what's our observed observation and what did we expect? What's a t-test? What's ANOVA? What's false discovery rate? And then we'll get into permutation statistics and then a few bit trickier ones. And in a lot of this class, we'll build on simple things that will snowball quickly into things you'll be able to understand at higher and higher levels of that. Oops, excuse me. So yeah, so we're gonna start interpreting the data, and that's what we did last year, and that led to data visualization, our bioconductor, identifying packages we may have needed based on what we found. So after module 5 is kind of like, let's explore and write a paper together. And that worked great using a lot of the online resources that we can do all of this virtually. Anybody can take this class with us if they want. We'll get into all of that in the next video, how we're going to do all of this virtually in one place, essentially. So that's kind of the outline. And another way of looking at this is we're going to learn Bash and Unix to go get data, use GitHub to collaborate and share code and work together as a group to analyze a ton of data. It's going to take maybe 10 days to run, but don't worry, we'll run it for you and we'll get there, but you guys will each run subsets of the data. Then we're going to really add in some statistics to make sure we're kind of getting on the same page. An initial statistics lesson. The syllabus, again, will be go over in the next video along with the other online resources. We use a virtual computing environment, workflow, and containers to make sure we can take a ton of data from one source, pipe it through a bunch of analyses, understand what those are intuitively, and then what statistics we would apply after the data has been aligned to the genome, outputted. What did we find out? What kind of statistics would we ask for our hypotheses? And really, after learning these modules, I think the data world's your oyster. You can find a container workflow or build your own container workflow. You develop one for whatever type of data you ever really are interested in. I think it would work for astronomy, to be honest, with just different pipelines. So really, there's in biology, we hear of all these things: a single cell seq, tag-seq, ChIP-seq, SNP-seq, PRO-seq, NET-seq, CRO-seq, RNA-seq. And really, Nextflow uses virtual compute systems in order to use all of the correct versions of these different bits of pipelines of analyses version tracks them all, has them all bundled, and you can put online a virtual compute node that anybody can literally take your FASTQs, put it through the exact same workflow, and get the exact same results. 100% reproducibility is our goal, not pseudo reproducibility, and we'll get into that into later lectures in the class. So really, it all can be bundled, and you can make these bundles for people if you get into this. So really, you just want to have a nice spot where you can take data from somewhere that's reliable, put it into a pipeline that's going to do all the necessary steps for you, and output all the necessary data that you want or that most people need. And then you can make more data from that, obviously. So yeah, so this is sort of the 6 modules we have. Classes pass fail. We really just want you to feel comfortable on a computer and feel comfortable on the internet, and even have some fun examples of how to download YouTube videos if you really want to from Unix and Bash, just to get some concepts down of how this will be relevant to you in your regular life. And then we're going to get into more of how is this relevant to the human genome in the second half. Some students will need to take it for grade, and we can discuss that in the first lecture. So module 1, we'll start with Bash. I think this was a funny little license plate. Bash or Unix is just such a great historical thing of how elegance and simplicity really were powerful. I mean, essentially put people on the moon. And it was developed by these people, and we'll go more into the history of it into lectures as we go. But they really wanted the terminal as essentially the first graphical user interface, or GUI, in order to talk to a kernel. Just as a fun fact, Linux is not a Unix operating system. Instead, it's Unix-like. Pretty much everything is Unix-like. I prefer just to use the word bash throughout the course, which is the sort of the pure forms of these commands. But to each their own. And really, the way this sort of works in a simplistic way is that you have your computer chip that has a kernel that can talk to it. And it's sort of, if you will, this level, privileged computer speak. And what we use is the shell, or the user speak. So we speak in a shell to the kernel, which is almost like if you think of a shell to the computer chip. And so we're getting really close to talking directly to the computer chip. The born again shell is the sort of the basic language of how would you talk in a way that to a kernel to make sure the instructions sent to the computer were optimal at best. And so really short bits of information, just absolutely elegant, and we'll get into a lot of those commands. The next video will show you how we are going to teach this in a practical way, just with the online resources, and just kind of a quick overview of it. But in Unix or Bash, you basically have a standard input into a program, which either gives you a standard error, and it'll display that, or a standard output, and you typically want to put this into a file. So it's just interesting to think that you put something into a command in Bash, such as grep, and it's thinking about it. It knows the answer in standard output, but it doesn't do anything with it until you tell it to print it out. And that can be really powerful by having the computer think about one thing, incorporate another aspect, with pipes in Unix, and we'll go over all that as well. And then you can access from your Bash terminal or your shell, tail, head, cat, print, nano. We'll go through several different ways of accessing files and looking at them in Bash. This is kind of important because you need it at some point to sort of get a bunch of stuff done on the server or somewhere. And you really can't get around needing to know some basics of Bash. We'll give you all the kind of core aspects of it. And this is just a fun example. I don't know if you can tell the difference between these two commands here, but in one case, you'll remove all the files in a folder. In one case, you'll remove the folder altogether. And it's just simply this space right here that would make all the difference in these two things. So we'll just give you some basics in the syntax, grep, awk, sed, and for loops in Unix is one of the first lectures. We'll be doing all of this on Fiji. We have an amazing bio IT department. These guys just are incredible, and they deal with all of the compute across so many different departments at BioFrontiers. They'll give a guest lecture on best practices on Fiji, but they're going to allow the class to proceed on the server. So essentially, you could use a phone for this class if you needed to. You don't need to have a hard drive. Everything will be on the server. Every command in Bash you do will go access another server. So you really could do this on a phone if you have really good vision and really fine typing skills. Our studio would be pretty tough, I have to say. But anyways, this is again just sort of the overview. And now the second sort of explaining into how we move into module 2 is we're going to learn about Encode. And Encode is cool because it started in 2004. I didn't fully get it. It turns out I'm an author, and I was ignorant to how important this effort was when it first started. I thought it was great that a bunch of scientists would work together, but I didn't understand how important data reproducibility and management of data and recording aspects of data and data sciences. As data expanded, this became more and more necessary. This was the first version of Encode in 2004. And in 2020, 16 years later, they had the fourth iteration of it. And so I've been very proud to be part of it, even though I didn't fully understand how important it really was. And so we're going to use this philosophy of data reproducibility that Encode sort of pioneered for biology throughout our class like we did last year. So we'll get into how to go get it, where is the data. Encode has an amazing API that they can even teach you how to program the API to get things you want. So it'll search for you for any sort of data you're interested in. Last year, we honed in on this. We'll go into this in the first lecture, actually. How do you find data in Encode? There's tons of it. This is outdated already. Last year we did K562. This, like I said, is outdated. HepG2, we're now going to do double that. I think 4 times as much data this year, twice as much data this year. Yeah, we're just chugging through cell lines and using pipelines, just like Encode's goal was to provide data in a very accessible way that's marked down, and all of these properties, we're going to show you how to grab them and make sure they're not issues with your data. We use CHIP. This is chromatin immunoprecipitation. Basically, simply, where do proteins bind on DNA? We have 3 billion letters in our human genome, and Encode was really smart to standardize not only the experiments, but also the data analysis pipelines, such that very similar types of data outputs would be generated. And so it would be sort of neutralize any artifacts across things. And if so, either way, it's well documented, so you can understand how different read lengths or other things affected your data. And so the basics of this is you break open cells and you cross-link them, and freeze them in time, break them open, IP the protein of your interest that binds to DNA, and you find the pieces of where it binds in the genome. Last year we did, I guess this is updated, we ended up with 195 DNA binding proteins, and this year we're going to shoot to understand 400. And the cool thing about this approach is you're understanding how a ton of different proteins are interacting in one cell, is what we did last year. And now we'll see how a bunch of different DNA binding proteins interact in a different cell, and ask, were they similar or different between those 2 cell types. So yeah, this year we finished K562 last year. This year we'll do HepG2, and there's way more data than this shows. But we'll also show you how to get the raw data. Not Encode's great, but not every data set has this API or ability to search the data in a great way like Encode does, and tools to download it all right away. But sometimes you just need to go to the basic sources where people submit their raw data after their publications, and we'll look at how to do that as well. And then what we really need to do is make sure we're keeping track of our collaborations. And sorry about the login here, but this just, GitHub is critical for collaborating, and we're going to be working in groups, and you'll each have your own branches, and we're going to work in the same GitHub, and try and mimic the environment you would typically work in, where you could work on any project, to be honest, in the world, and contribute to it. How do you do that, and how are we going to do that together to separate tasks for analyzing the data? This is our GitHub from last year. It's a great way. We still have 10 contributors still active, I guess, left over from last year. But you'll see, we'll go through that in the next video on the supplementary sort of tools and resources we're going to use. But essentially, the whole class from last year you could reproduce in that analysis folder. And if you're interested, you can go to Boulder Rinn Lab Class 2020 GitHub and look at the knitted HTML files of all the analyses we did. All the figures are printed out. Again, this idea of why Markdown and reproducibility is so important for reproducibility. We print out all the figures in the Markdown files and turn them into HTMLs that anybody can download, get the code, and see the resulting figures. So we'll be working there together. We'll also use Slack, again, next lecture, but to supplement this. And after the first sort of flow of this, we'll go into after getting the bash and how to go get data. We're going to understand these pipelines, and it's actually quite simple. With 10 to 12 lines of bash code, you can invoke Nextflow to do days and days and days and days of computation for you by just giving it a list and a design file. So we'll go over how to make a design file for a pipeline, what's in the pipeline, what are the basic statistics in there. We'll focus on ChIP-seq, but we'll also get into RNA-seq, PRO-seq, and you'll see at some point there's so many commonalities that this is really quite simple. It says, where's my data? Where's the data you want? Here's the pipeline I know. Where do you want me to put it? And it gives you everything you pretty much need. So we'll go into Nextflow, how it works, but it's just basically a driver of how to manage a bunch of analyses simultaneously by marking down the versions, the outputs, and knowing exactly what happened at all times and recording it for complete reproducibility. So we'll go into that, and specifically the Nextflow pipeline looks complicated here, but this is really not that bad. And we'll teach you sort of intuitively what's happening. This is sort of like inside of a Nextflow pipeline. What are all the different steps? What's happening? What output files did Nextflow make for you? It did a bunch of analysis along the way. What are the output files and file types and things like that? So you have an intuition. Again, this will be applicable across any type of things. The file formats are usually the same. So that's Nextflow. We'll be doing the majority of our coursework for the class, and then we will move to analyzing the data. So we're going to do more with grep and awk and git and data file formats, understanding the algorithms and running the data. And file formats can seem confusing, but I think by the end of the class, you'll see we're going to use a few different types, and they'll be universally applicable across any types of sequencing platforms. The application, probably the best part of this whole thing is it's inquiry-based. So we don't know what we're going to get. Last year, we had no clue what was going to happen. Thanks to COVID, actually, we did learn how to do this online and with all these extra resources. And it made it actually better. So that's why we're keeping it in virtual format for now. And the application, just to run you through what we did last year, is download all the data we could that at least had at least one replicate, at least 100 base paired end reads. Don't worry about these terms, we'll get into it. But basically, how do you go out in those first 3 modules? How do you go out and get 1,000 data sets, throw them into a pipeline, and get output that you can analyze? So we did that, and we did a bunch of reproducibility aspects and data quality aspects that filtered out a lot of proteins. And we started with 195, we ended up with 161. But I think it was a good exercise in seeing what can actually go wrong, even with really high quality data. Here's just an example of some of the initial plots students were able to sort of generate, where we took the approach of if there was multiple replicates for a given protein, we collapsed them into one consensus. So when we say 161 proteins, that means they have at least 2 replicates, though, that were merged into one consensus peak. How do you do that? All of this is in pre-cooked markdown files that anybody could download. And we found right away something just sort of interesting, that upstream of this gene here that starts here, you can see the polymerase that turns this gene on is localized right on top of it. There's upstream of it, or 20,000 bases up from it, there's sort of an enhancer region here, where we brought in RNA-seq and pro-seq and histone modification data that indicated this might be an enhancer region that has enhancer RNA coming from it, and these transcriptional factors might be regulating it. So just right off the bat, pretty much right out of the pipelines, we were already able to sort of look at this level of data. And then we sort of use inquiry-based application to start learning some more statistics, like hierarchical clustering, to analyze how do all these proteins look? Do they look like they should? Here in red, we can see that they did, that the polymerase, these are all in a similar complex. Same with these guys. This one is even tagged, and it's similar to the one that was not tagged. We'll get into all of this as we go. But what was concerning, as we saw a bunch of data here, different proteins, totally different functions, but they all clustered, or all had more statistical similarity to each other, which is concerning, that might be the tag and not the actual protein. And then we sort of moved a little bit deeper into statistics in R, where we make plots like this, where we brought in RNA-seq and said, well, it's known that the more DNA binding proteins that are on a given gene, the more that gene will be activated, or the more RNA will be produced. So on the y-axis here, you're seeing the amount of RNA, and the x-axis, the number of DNA binding proteins, and in both different types of genes. We'll get into this in the class as well. But in both cases, they had a linear binding to that. So that was great. We could just do all this data analysis with the pipeline, and then we get out an answer with a real quick test that was already known. So the statistics, I like this quote from Fisher. I'll let you read it, but we'll go over quite a bit of different aspects of statistics. But really, we don't want you to have to know how to code them or math them, or understand the math completely behind them. We're going to give you just enough detail to know what to dig into, and what considerations you should take with the basics of your data. Like, is it a Poisson distribution or a binomial, right? You know, how is your data looking, and how do you use false discovery rates? And really what we like to do at the end of the class is use permutation, where we make null distributions by shuffling data and permuting it, and learning that permutation, shuffle, statistic test to see if it's significant, the observed versus expected. That can be used in any type of data analysis too. So we really try to focus on things that will be most useful in all types of analysis. We're also going to show you how things can look great until you think about them a little deeper, and why these permutation statistics and false discovery rates are kind of important. You'll end up throwing a lot of data in the trash. And then we're going to step it up a little bit, and with another inquiry-based application with R and Bash to use these permutation analysis I was talking about. And the application we'll use here is repetitive elements in the human genome, where the human genome has been constantly bombarded by transposons, or viruses, essentially, that integrate into our DNA and make us all very different. And there's all kinds of different old viruses that have attacked us, new ones, and they're all sort of recorded in the human genome. And so we're going to take a look at this, which is a great application for this permutation-based statistics. There's so many of these things that you can see any of them by chance. So we want to develop null hypotheses for the number of them in a family, and the size of them, and all sorts of aspects that you would want to consider for any element in the human genome. Transposed elements just make a good, I think, test exercise A, because we found something fun, and two, they're a great way to see how you can get false answers if you don't use permutation based statistics. Speaking of which, this is a very active area of our research where David Kelly found a whole new class of transposons that was later or worked with Ed Chung and Cedric Pashodi, who found the same thing genome-wide. And around 2011, there was this explosion of like, oh my goodness, these transposable elements are not just completely junk viruses, but there's certain ones that are critical for human pluripotency that are not in other species. So they're human specific and they're very highly regulated in pluripotent stem cells. And we still all work together, and Ed Chung is here and still works on this phenomenon as well. And last year we found something we thought was quite interesting, the newest transposon ever to be introduced to humans. So the last virus we've seen other than COVID and these other kind of things, it's a different type of virus. But these transposons, the SVA family, was very unique in the sense that most transposon families either bound everything or nothing. And that's why permutation analyses are really important, is that they don't usually bind anything. So if you saw one event, that would be statistically significant, but it would not be statistically significant in a permutation analysis. That was the goal. This one popped out, and it turned out to be really interesting that these three proteins bind adjacent or on the SVA, and in ProSeq and RNA-seq are actually expressed, and again have these marks of enhancers right next to them. So what it looks like is this SVA might be a repressor of enhancers or an insulator to enhancers, and or activate enhancers. We don't know. We looked in deeper as another inquiry-based application into sort of meta-analysis. How do you make meta plots? This is a really powerful aspect of R, I think, in plotting these features. And here we can see that indeed at a meta level, when we look at all SVAs, we can see that the K4 monomethylation doesn't matter right now. Let's say call it an enhancer. This protein 1, protein 2, and protein 3 are all bound within these SVAs, and they're strongly enriched to only bind in these things, whereas other modifications are not specific to this at all. So then after we had gone through all that and the students were getting proficient in R, that we sort of took it to another level where we noticed something bizarre along the way. That again, if we were looking here on the y-axis at how expressed the gene is versus the number of genes bound, this was that linear trend we had seen before, that the more things bound, the more RNA that's produced. But what struck out to us in the class is, how do you have like 110 proteins bound at your promoter, but no expression? So this is where we started to meld skills and work together as a group and try and figure out what new things are we going to need to bring into this data set to better understand this. And so we started to call these reservoirs, where there's reservoirs of DNA binding proteins bound on a promoter, but there's no product being produced by RNA-seq at least. So we looked at ProSeq, which is nascent transcription, if there's any hint of activity going on in there. And we did, indeed, we saw that these things do look like enhancers, because the amount of RNA that's coming out of them at nascent levels at least looks similar, that has a similar trend, that the more proteins that are bound at the promoter, the more nascent transcription there is. Any of this stuff doesn't really matter. Just you'll learn how to plot these things, what statistics to run, and how to go after something that's unknown and newfound. One concern is that these reservoirs might be super enhancers, so we brought in data from other places that said, no, they're not super enhancers, they're very different. A lot of properties. This is just one of the plots in the paper. And really, we just sort of spent a lot of time working with students to plot the figures of their interest that they wanted to sort of highlight in the paper, and got to the point where we were ready to sort of integrate this. This is some examples of all the visualization you can do in R. It's quite powerful in the last module. And we ended up having enough time to get into science writing. How do we incorporate these results? We submitted a paper to bioRxiv that's just recently gotten good reviews from PLOS One. And we chose PLOS One because the goal of PLOS One is to have findings that are highly reproducible and less about novelty. I think some of the findings we had were interesting and somewhat new, yet the idea was the reproducibility of this, that you can pipe this whole class and get the exact same paper, was really the main goal. And the students got to see that come full circle. And we're finishing up with the reviews now. But anyways, I just thought this was nice what the reviewer said here. And this in the class last year got a really good score. I think because we all worked really well as a group, and everybody was learning together, even Michael and I. So we're hoping to do the same thing this year. I think it should be more streamlined. We know some things that didn't work. For instance, we think we didn't introduce R quick enough last year, and sort of the students were struck by R coming in so strong near the end. So we're going to work on integrating that and teaching you the basics of R in an inquiry-based way, where you kind of want to know what the answer is, and data you would typically need to know what the answer is. So in the next video, we'll go through what those R markdown exercises look like, that will all run together in Fiji, the same directory, and work on the same GitHub together. So let's do that and explore hundreds of DNA binding proteins across the human genome. I believe it's 444 this year. Don't quote me on that, but it's going to be way more than 195. And we're looking forward to seeing what we see this year. And hopefully, we're pretty sure by the end of the class, no matter what, you will understand what it's like to be a data scientist. And that's really all the goal of this class is, what are the terms, basic functions, ways of thinking that will get you to be able to not only access data and analyze it, but do it in a way that you're making sure anybody can reproduce it. And that's becoming a very, very important aspect of every type of science nowadays. So with that, we think you can. We did it last year. We're hoping it'll be even better this year. And in the next video, I will go over the online resources we're going to use, how we're going to use them, what some of the lectures look like, what is this going to look like, how do you learn to do this on a computer virtually. And that's kind of part of the fabric of this class, is that we really think with these resources in the next video, anybody can take this class. We just won't have as much time to work together about what do we want to write a paper on, or what do we find interesting in the data. But all the fundamentals will be pre-cooked and available and runnable prior to class. So we're looking forward to it, and we'll see you there. Bye.